Which of the following interactions is primarily responsible for the stabilization of an alpha helix in proteins? A. Hydrophobic interactions. B. Hydrogen bonds. C. Disulfide bonds. D. Ionic interactions. The correct answer is B. Hydrogen bonds between the carbonyl oxygen of one amino acid and the amide hydrogen of another, 4 residues away, stabilize the alpha helix structure in proteins. Which of the following is not a function of carbohydrates in biological systems? A. Energy storage. B. Structural support. C. Signal transduction. D. Catalysis of biochemical reactions. The correct answer is D. Carbohydrates serve roles in energy storage, structural support, and cellular signaling, but the catalysis of biochemical reactions is a function primarily performed by proteins, enzymes. Which enzyme is responsible for the unwinding of DNA during replication? A. Topoisomerase. B. DNA polymerase. C. Helicase. D. Ligase. The correct answer is C. Helicase unwinds the double-stranded DNA by breaking hydrogen bonds between complementary base pairs, allowing replication to proceed. What is the primary driving force behind the folding of globular proteins into their tertiary structures? A. Ionic interactions. B. Hydrophobic interactions. C. Disulfide bond formation. D. Van der Waals forces. The correct answer is B. The folding of globular proteins is primarily driven by hydrophobic interactions, where non-polar amino acid side chains are buried inside the protein, away from the aqueous environment. Which of the following types of RNA is responsible for bringing amino acids to the ribosome during translation? A. mRNA. B. rRNA. C. tRNA. D. snRNA. The correct answer is C. tRNA, transfer RNA, carries specific amino acids to the ribosome, where they are added to the growing polypeptide chain during translation. Which of the following fatty acids is classified as an omega-3 fatty acid? A. Palmitic acid. B. Oleic acid. C. Linoleic acid. D. Alpha-linolenic acid. The correct answer is D. Alpha-linolenic acid is an omega-3 fatty acid, characterized by having a double bond 3 carbon atoms away from the methyl end of the fatty acid chain. Which statement best describes the role of cholesterol in biological membranes? A. Cholesterol increases membrane fluidity at low temperatures. B. Cholesterol decreases membrane permeability to small ions. C. Cholesterol forms the lipid bilayer of the membrane. D. Cholesterol acts as a channel for ion transport. The correct answer is B. Cholesterol modulates membrane fluidity and reduces the permeability of the membrane to small, charged molecules by inserting itself between the phospholipid molecules. Which type of bond links monosaccharides in a disaccharide or polysaccharide? A. Peptide bond. B. Glycosidic bond. C. Phosphodiester bond. D. Hydrogen bond. The correct answer is B. Monosaccharides are linked together by glycosidic bonds to form disaccharides or polysaccharides. These bonds form between the hydroxyl groups of adjacent sugar molecules. Which of the following amino acids contains a sulfur atom in its side chain? A. Serine. B. Methionine. C. Valine. D. Leucine. The correct answer is B. Methionine contains a sulfur atom in its side chain, making it one of the sulfur-containing amino acids along with cysteine. Which of the following best describes the function of ribozymes? A. They are proteins that catalyze reactions. B. They are RNA molecules with catalytic activity. C. They are involved in protein synthesis only. D. They are part of the electron transport chain. The correct answer is B. Ribozymes are RNA molecules that have enzymatic activity, meaning they can catalyze specific biochemical reactions, such as the cleavage of RNA during splicing. Which of the following molecules is a precursor for steroid hormone synthesis? A. Glucose. B. Cholesterol. C. Phosphatidylcholine. D. Acetyl-CoA. The correct answer is B. Cholesterol is the precursor molecule for the synthesis of steroid hormones, including cortisol, testosterone, and estrogen. Which of the following nucleotides is found exclusively in RNA and not in DNA? A. Thymine. B. Uracil. C. Adenine. D. Cytosine. The correct answer is B. Uracil is a nucleotide that replaces thymine in RNA. Thymine is found in DNA, while uracil is specific to RNA. Which coenzyme is required by the enzyme pyruvate dehydrogenase in the citric acid cycle? A. FAD. B. CoA. C. NADP jod. D. Biotin. The correct answer is B. CoA. Coenzyme A is required by the enzyme pyruvate dehydrogenase to convert pyruvate into acetyl-CoA, which enters the citric acid cycle. What is the primary function of chaperone proteins? A. Catalyzing peptide bond formation. B. Facilitating the folding of other proteins. C. Degrading misfolded proteins. D. Transporting lipids across membranes. The correct answer is B. Chaperone proteins assist in the proper folding of other proteins, preventing aggregation and ensuring they attain their correct functional conformation. Which lipid-derived second messenger is generated by the hydrolysis of phosphatidylinositol 4,5 bisphosphate PIP2? A. DUG, diacylglycerol. B. CAMP. C. CGMP. D. ATP. The correct answer is A. Phospholipase C hydrolyzes phosphatidylinositol 4,5 bisphosphate PIP2 into two second messengers, DUG, diacylglycerol, and IP3, inositol trisphosphate, both of which play critical roles in cell signaling. Which class of enzymes is responsible for catalyzing the phosphorylation of proteins? A. Phosphatases. B. Kinases. C. Ligases. D. Hydrolyses. The correct answer is B. Kinases are enzymes that catalyze the transfer of phosphate groups to proteins, typically on serine, threonine, or tyrosine residues, regulating their activity. Which of the following amino acids is considered essential, meaning it cannot be synthesized by the human body? A. Glycine. B. Alanine. C. Leucine. D. Proline. The correct answer is C. Leucine is an essential amino acid that must be obtained from the diet because the human body cannot synthesize it. What is the main role of ATP synthase in cellular respiration? A. Breaking down glucose into pyruvate. B. Producing ATP from ADP and inorganic phosphate. C. Transporting electrons in the electron transport chain. D. Pumping protons across the mitochondrial membrane. The correct answer is B. ATP synthase is an enzyme that synthesizes ATP from ADP and inorganic phosphate, utilizing the proton gradient generated by the electron transport chain during oxidative phosphorylation. Which of the following polysaccharides is a major component of the plant cell wall? A. Glycogen. B. Cellulose. C. Starch. D. Chitin. The correct answer is B. Cellulose is a polysaccharide that serves as a structural component of plant cell walls, providing rigidity and strength. Which of the following biomolecules acts as a carrier of acyl groups in fatty acid metabolism? A. Coenzyme A. B. ATP. C. NAD. D. FADH2. The correct answer is A. Coenzyme A carries acyl groups during fatty acid metabolism, playing a key role in the synthesis and degradation of fatty acids.